Oxford Brain Diagnostics is a biomarker group, so I wanted to emphasize a little bit about biomarkers in this discussion. So here's the way the FDA approaches it. Biomarkers are in one of 7 recognized contexts of use. You guys work in this area, so you probably know this, but not everybody does. So a biomarker must be in one of these areas, and a biomarker would always be defined as having a context of use. So as you see there, it's either a risk, a diagnostic, a monitoring, a pharmacodynamic, a predictive, a prognostic, or a safety biomarker. And one biomarker might have several contexts of use. For example, you could use amyloid PET for diagnosis, and you could use it as a pharmacodynamic biomarker for amyloid reduction. So that's common, but both of them would have to be declared, and both of them would have to be qualified, that both context of use would have to be qualified, and there's a qualification process through the FDA that allows a biomarker to become qualified with these. So I'll just summarize what I think is important and what I've been thinking about, and really looking forward to the discussion. There are many mechanisms represented in the pipeline. I've emphasized inflammation and synaptic plasticity, but you see that virtually every CADRO classification is represented. The biomarkers are enormously helpful. I just showed you Trailblazer as one example. There are many examples. I think the idea of doing drug development without biomarkers is almost unthinkable at this point. But we need a lot more. You think of the thousands of cellular processes that are involved in Alzheimer's disease and how few biomarkers we have compared to that. So we need a lot of work.
Thank you very much, Jeff. For a wonderful, comprehensive review, actually, and also very thoughtful. Thank you for thinking a little bit about how Oxford Brain Diagnostics tools might fit into that. I mean, not only obviously were your direct references useful and food for thought for us, but I was thinking also about in terms of groupings of drugs, those that are targeting the synapse. And you mentioned effectively the kind of integrity of the circuits. I think that's quite interesting because although you referred to functional types of assessment, fMRI and EEG, in many ways I think we consider that we're assessing neurodegeneration and we're looking for the structural corresponding features. So because we're looking at microcircuits, you know, our target in the original design of our method was around the mini column structure, which is the ubiquitous sort of circuit feature of the cortical layers. So I think that's very interesting that we might particularly apply ourselves to that area. But of course, a marker of N in the ATN framework is very much where we see ourselves sitting. So in multiple ways, I think we are relevant to this picture. But that's really just a comment, a bit of positioning, if you like. But I'm not anticipating too much about which questions will come because the whole team will, I'm sure, have many questions. Who would like to ask?
I'll just get back to that, Stephen.
Yeah, sure.
I wondered about that when I looked at the website, whether your measures would tell us something about circuitry, because you probably know that it's been very difficult to do fMRI at the multi-site level and have it be really solidly reproducible.
Yeah.
So if we had a structural measure that we could use instead of fMRI, it would have a great advantage.
Yeah.
So I encourage you to keep working at that, because we would love to have measures that, you can put, what the, I don't know, 140 sites in some of these trials, maybe even 200 sites in some trials. Well, that's ridiculous in trying to do fMRI. So an alternate measure would really be valuable.